Hello, my name's Shaun Robertson and I'm going to talk about polymicrobial biofilms and the importance of building better biofilm models for research translation. So I'm a research fellow from the National Biofilms Innovation Centre at the University of Nottingham in the United Kingdom. My work has focused in the last few years on building this area and just building models that will help translate research from the university environment into academic. Sorry, I'll start that again. Right, okay. So, hello. My name's Shaun Robertson, and I'm a research fellow at the University of Nottingham in the United Kingdom. I'm going to be talking about polymicrobial biofilms and the importance of building better biofilm models for research translation. So when we think about microbial biofilms and why they are important, it's not just that they are involved in healthcare, there is a wider impact in the industrial area as well. Often with these microbial biofilms, they are microbial in nature, and in this terminology, that means more than 3 species of microbes are involved in forming that biofilm. And these have a large range of effects in healthcare with morbidity and mortality, and in industry with a large economical burden as well. And this has been quantified recently in a report in NPJ Biofilms and Microbiomes, with the economic significance of biofilms by sector being evaluated. And this has grossed over nearly $4 trillion globally, with a large component of this being due to microbially influenced corrosion. When we exclude the microbial influenced corrosion, as you can see on the right-hand side of this pie chart, there's a significant medical and human health issue, but also personal care, oral health, home care, and so on and so forth. And focusing in on the economic significance of these in the healthcare sector, we can see that there's a large impact across a number of areas that some of you may be familiar with, especially those being highlighted being things like wound healing, which is one of the largest economic impacts globally, and also other diseases such as the orphan disease cystic fibrosis, which is typified by microbial infections, especially later in adulthood, predominantly through a bacteria known as Pseudomonas aeruginosa. And when we represent this as a kind of overview of the sites of infections in humans, you can have the oral cavity, which is a very well described and literature-rich area of the investigation of biofilms and polymicrobial biofilm models, and then into other areas such as cutaneous sites, vascular catheters, intra-abdominal sites, UTIs with catheters, and also lower reproductive tract, burn wounds, and the respiratory area as well. So when we talk about a biofilm, the conceptual model, which has been really well illustrated in this recent review, is where we have a surface, microbe comes along, colonizes that surface in what is known as a reversible attachment, and this is seen on the very far left of this schematic. Then we have the transition to irreversible attachment, production of extracellular polymeric substances with cell clustering, which essentially glues it to that surface, and this is the beginning of what we call a biofilm and maturation. And then into the microcolony formation and then dispersal through various methods, whether that's active or passive. Yet, when we look at the conceptual model, which we can replicate in lab, in vitro, this is not the case. And this has been typified when we look at flow in open systems, where we, instead of seeing this heterogeneous mass of biofilms that form with structures that are mushroom-like in cases and also have these pellicles forming out of water streams. And this is in an open flow system rather than the traditional flow systems we see in labs. And then we look at cystic fibrosis, which I mentioned earlier. We see these self-aggregated biofilms where they're not actually attached to a surface, which has normally been a typical definition of what biofilm is. And then looking at another area, wounds, where we see these biofilms that are polymicrobial in nature and typified by some attachment to areas, but then also present in the wound exudate as well. So this leads to the questions of how do we better translate research and how do we better mimic these? Well, what we need to do is we need to consider a number of factors. And this is very well summarized in a review by Peleg, where we need to look at how these microbial species mix. Are they mixed? Are they segregated? Do they layer? And do they change over time? And with those chemicals, byproducts, metabolites, are these mixed, segregated, diffusible, dispersible? Are they metabolized by other microbial species? These all need to be considered when looking in context of polymicrobial biofilms. And then this can also have other effects where you can have physical interactions, such as this typified here. You can have a fungi yeast body with a hyphenating cell, such as Candida albicans. And then you can have attachment of microbes that can be either positive or negative for the fungal interaction. But then also these interactions can modulate the host response as well. So this is a complex environment with complex interactions, yet traditionally we have tested on single species. And just to give an idea of what can happen in the cystic fibrotic lung with patients that are colonized by a number of microbial species, such as Burkholderia, Stenomorphus, Pseudomonas aeruginosa, and Candida albicans, they produce a number of molecules that are used in cell-cell communication, and these are known as quorum sensing molecules. So what we can see is there's a number of impacts where we have Burkholderia having negative inhibitory impact on the quorum sensing biofilm formation and virulence of Pseudomonas aeruginosa. But then we can also have things like Pseudomonas aeruginosa producing its quorum sensing molecules. They can negatively impact the filamentation, so the formation of hyphae by the yeast body of Candida albicans. And conversely, Candida albicans can produce a compound known as farnesol that can then impact on the quorum sensing of Pseudomonas aeruginosa. So it's quite evident that there are complex interactions that are known in the literature that need to be taken into account when we model these microbial species. And just to give some further real-life examples, we have this where the dental biofilms are a very well-established area where we know the interactions, and there can also be hierarchical structuring of the early bridging organisms which attach to the dentin surface of the tooth, followed by attachment of secondary and tertiary colonizing microbial species. And generally what happens is it's the tertiary microbial species, such as Porphyromonas gingivalis and Fusobacteria nucleatum, that cause things like bad breath and other oral-related disease, such as gingivitis. And then when we look at infection, especially in systemic infection, we have Staphylococcus aureus cooperating with Candida albicans. This is the SC5314. What we can see here is the invasion of mouse tongue tissue by the Candida albicans hyphae, and the Staphylococcus aureus attach to these and travel with the hyphae. Yet, when we knock out a protein expressed on the surface of Candida albicans called ALS3, we see that Staphylococcus aureus does not then attach and does then not invade alongside the Candida albicans invasion. And then when that is recomplemented into the knockout, we see that that function is restored. So it's quite evident that there's a lot of interesting cooperations, synergisms, and antagonisms between microbes and how they function. And this is really important when we're looking at how we model biofilms. So how do we go about modeling biofilms? This is really well summarized in this infographic here, where we have what is seen as essentially a continuum of how we model biofilms, from the very, very lab-focused stage on the left-hand side, which are normally seen as very cost-effective, high throughput, quite easy to perform, yet they are very poor mimics of the in vivo infection. And then this goes all the way through to the more complex models, such as the invertebrate kind of in vivo models, non-mammalian ones, human organoid models, which have had varying success and are a really interesting area of current research and will have great application in the future. And then going on to the mammalian models, which are expensive, very low throughput, and also have ethical implications. So to delve into these a little bit more, just to give some examples of these, with in vitro we can go from basic microtiter plate assays through to the Calgary biofilm device, which has been around for about 20 odd years now, and has really helped in the research of Pseudomonas aeruginosa through to colony biofilm models, which is what we predominantly use for our basic research and investigation of polymicrobial interactions. And then what this then allows us to do is build up basic research and science understanding to then go on to more complex models. And more complex models, such as these, can look at the Lubbock wound biofilm model, the collagen model, and this is essentially designed to mimic a grade 1 diabetic foot ulcer, and then into other models such as the microfluidic platforms, which can then be used to look at biofilms microbiology under flow conditions, such as in a catheter, for example. And these then allow us to move into other models, and I would say that there's a kind of bridging between the in vitro and in vivo, and that's the ex vivo biofilm models. And these generally are performed on porcine ex vivo samples, such as the porcine skin model, which has seen great use in the last few years, and also the porcine lung model, which is used in some ways to look at Pseudomonas aeruginosa biofilm formation and treatments in a CF-like condition. And then moving on, we have the in vivo models with the invertebrates, such as Melanella galeria or the greater waxworm, and these can be used to monitor biofilms growth, especially if you have things like a Lux reporter, as can be seen here. When you have biofilm in formation inside the waxworm, you can see it through the waxworm. And then also moving into things like C. elegans as well, where you can use things like GFP reporters as well to visualize the microbes. And then moving into the murine area, we have the complex biofilm models, such as this one here, which is one that we use at the University of Nottingham, which is a subcutaneously implanted infection site, and then which is allowed to heal, and then the infection is monitored longitudinally using an IVIS, and this work is performed by Dr. Jenny Luckett. And this allows us to do very high throughput longitudinal monitoring, so with the same mice, which has greatly reduced the use of mice in our research and is also concurrent with the views of the NC3Rs in the UK. So it's quite evident there's a number of biofilm models. We need to better translate these, but we also need to do the basic research to then get to that stage. So what we've been looking at is first obviously understanding how polymicrobial biofilms can interact, and the best way to do that was to go back to the start. So what we've done and spent quite a few years working on in standardization of a polymicrobial colony biofilm model and making sure that it's reproducible in a number of labs, including collaborators in America. So this model is very simple, but is quite complex to set up in the first place, as it requires making sure that the microbial species here are added at the right point. And in generally, what we found is on initially doing this work that we would have complete overgrowth of Pseudomonas aeruginosa. Therefore, what we've then designed to do is to lay this addition to then get a stable biofilm, which can then be used, for example, to test novel compounds, but then also look at testing things like wound dressings. And this can have a number of endpoints, such as the standard colony forming unit analysis, or looking at things like viability imaging, so live dead confocal imaging, and then looking also at things like viability PCR to then establish live dead populations through PCR. So what we've done with that is then apply this to a number of models and areas, such as the visualization of how these biofilms form. So what we found is there's actually a spatiotemporal formation to these. And what we can see here is the Candida albicans, which is in the green in the center of this image, and then around it in magenta is the Staphylococcus aureus, and it clusters around it, and on the edge of the colonies of Candida albicans we can see hyphenation. And then after the addition of Pseudomonas aeruginosa at 24 hours, we can see the integration of Pseudomonas into that biofilm structure. We've seen maturation of the Candida albicans in green, the magenta, which is the Staphylococcus, clustered around that, and also in and around the hyphae of the Candida albicans. And the Pseudomonas is starting to make headways into those structures, as is seen in that middle image with Pseudomonas represented in cyan. And then what we see at 72 hours is a disruption of that initial tertiary structure. And then what we see now is a homogenization, almost, of the Pseudomonas throughout that biofilm. A lot more hyphenation with the Candida and the Staphylococcus aureus distributed throughout. And this is quite a stable biofilm structure that we've managed to grow for a number of days past this point. But then when you also look at these polymicrobial biofilms, they look like a biofilm, but it's a colony biofilm model. So we need to make sure that it is definitely a biofilm. And to do that, we've done carbohydrate staining and also looking at extracellular DNA, which are both hallmarks of a biofilm formation. So at 48 hours, we see this kind of biofilm structures, and you can see in the top left, there's some evidence of what is one of these Candida albicans, yeast, and hyphal structures. But then when we move to the 72 hours, and then we see these distributed change in the biofilm architecture in green, and then also with the distribution of the extracellular DNA seen in blue. So we know we now have a biofilm in this polymicrobial biofilm. And then another thing to always consider as well is the environmental conditions under which the tests are conducted. And this is really exemplified by a paper that was published earlier this year, which showed the impact of the environmental oxygen conditions, where we have normoxia, which is classed as 21% oxygen, and hypoxia, 1% oxygen. And this is a really strong driver for the filamentation of Candida albicans, so moving away from that yeast growth morphology towards the morphology of forming hyphae. And this was really interesting work that allowed them to then unlock the molecular mechanisms by which this is regulated. And as you can see here, there's quite a stark difference between how the Candida albicans grows under normoxic and hypoxic conditions. And this is important to consider when we look at translational research. So another thing that we're able to then do is look at, well, what about standardized testings for the translation of research? So standards, claims for antimicrobial activity. And a very easy one to focus on is that of wound dressings. So this is generally regarded as an antimicrobial fabric. So one of many tests that can be used to claim antimicrobial activity is the AATCC100, and it can be used to test for this. And the standardized methodology that is available for people to conduct and to provide a claim for antimicrobial activity. Yet when we look at this, it tests versus planktonic bacteria. Yet this is not the preferred growth mode of microbes, which is in the sessile biofilm state. Yet there is no requirement for testing versus biofilms in this standard. When we know that this is dominant, and we know that these biofilms are also not just monospecies but polymicrobial in nature. So using the colony biofilm model that's polymicrobial, I was able to show that while you have these monospecies biofilms, treating them with chlorhexidine, which is used as a topical antimicrobial, especially for decolonization before surgery for Staphylococcus aureus predominantly, we see that on a 1% solution, which is below what's used surgically, that we have reductions from the control to chlorhexidine 1% of about 2 logs with Pseudomonas aeruginosa, a 5 to 6 log reduction with Staphylococcus aureus, and a 2 log reduction with Candida albicans. Yet when we shift into this polymicrobial model with Pseudomonas, Staphylococcus aureus, and Candida albicans in SC53114, we see that there is a greater recalgrance to that treatment with 1% chlorhexidine. And this just exemplifies why testing in a polymicrobial nature is very important compared to just testing versus microbes on their own, especially in biofilm states. So then another study that we then followed on with this after validation of that model and seeing this interesting effect was to look at the impact of currently available commercial wound dressings represented by 1, 2, 3, and 4, and on the impact of this polymer microbial model. And it was really interesting to see that when we performed this biofilm challenge with the whole intact biofilm, we didn't see any viability impact with 3 of the dressings. Only 1 of the dressings had an impact out of those 4. And this was also seen to a lesser extent when we also looked at disrupting those biofilms and then challenging the wound dressings, where we still saw a very high level of recovery of the microbes. So what that's led us to is allowing to look at this translational aspect, look at why treatment may fail, why wound dressings potentially may not translate from the planktonic standard testing to killing biofilms. And then it's also allowed us to look at how we can harness new technologies to look at investigating how these microbes talk to each other in these polymicrobial biofilms. So looking at Pseudomonas aeruginosa as our organism of choice at Nottingham, Pseudomonas aeruginosa is regulated by 3 quorum sensing systems. So we have the LASR and the RHLR, which are N-acyl homoserine lactones, so AHLs. And we also have the Pseudomonas quinolone signaling PQS, governed by PqSR. And these 3 systems work in tandem to regulate the Pseudomonas aeruginosa and its virulence determinants. So one of the things that we've used these models for is looking at how we can modulate that interaction, that regulation, and also what would make effective targets to reduce virulence. And one of the ones that has been actively looked at is antagonists of PqSR, so knocking out the PQS signaling in Pseudomonas aeruginosa, which has a number of beneficial effects. Being able to see is with the PQS, knocking out the PQS with this PqSR antagonist in this paper that we published last year, the part of this pathway is formation of compounds such as HHQ and PQS, which are involved in the biosynthesis pathway of Pseudomonas aeruginosa and its regulation. And what we're able to see is that when we add this antagonist, we're able to knock out the production of these molecules in most cases, but not all. Some of them are resistant to this, and these are the essentially representatives of the 5 clades of Pseudomonas aeruginosa. But more importantly, what we can do with this is looking at combination of the quorum sensing inhibitor. So here we have what we call 61, and in combination with antimicrobial that is commonly used, such as ciprofloxacin, we're able to, at early points in the biofilm, essentially sensitize it again to the antibiotic. This is quite important early research, and it also has downstream impact as well, because when we knock out these quorum sensing systems, we can impact things such as pyacinin production, which is seen as a virulence determinant in these infections. Yet when we're doing this research, we don't exactly have the capability to see into these biofilms and also to map the distribution of these chemicals, metabolites that are in conflict between microbial species and also between the host as well. So what we've been doing is validating and standardizing on a new bit of equipment at the University of Nottingham that's also present in NPL down in London, and that's called the 3D OrbiSIMS. So this is essentially a very advanced method of mass spectrometry. It has high spatial resolutions for biomolecules, and it can also be done in 3D to pinpoint where these are in complex samples. And there's a number of modes that this can be run in, of special importance being profiling. So we'll be able to drill down through a biofilm and see the structure inside it, and then also to build up 3D imaging once the initial experimental validation data has been obtained. So what this looks like is we have the biofilm in the top left pre-measurement, and then we have post-measurement where we then expose it to essentially an ion beam that will etch off the biofilm and put it into the mass spectrometry for analysis. And from this, we've been able to detect a number of the key biosynthetic compounds in the PQS synthesis pathway, such as the C7PQS, C9 quinolone, C9 PQS. And from that, you're also able to get a distribution through the depth as well. And just for clarity, the indium is part of the coating on the slide, the indium tin oxides, which helps with the signal to noise ratio for the OrbiSIMS analysis. So we've been able to work through this and then look at it in a dual species nature at the moment, where we've been able to establish levels of compounds that are in the biosynthesis pathway, such as HQ, HQNO, which has interactions with Staphylococcus aureus, NHQ, and PQS. And what's quite interesting is when we then look at this in combination with Staphylococcus aureus, we're able to see a reduction in the levels of these compounds and also the impact that it has on the Staphylococcus aureus through other visualization methods such as confocal microbiology. So hopefully we'll be able to use this further downstream to then look at producing 3D imaging maps and then also working at quantifiable concentrations from these imaging techniques such as the OrbiSIMS. But this is where it helps us to also analyze other complex samples such as patient samples, when we'll then be able to take that, analyze it, and tell what microbial species are present through hallmark chemical species such as things like HHQ, PqS that Pseudomonas produce, or AIPs for Staphylococcus aureus. So at the end, it comes down to what we need in the area of biofilm research is the understanding that there is no perfect biofilm model. What it requires is for people to use the right biofilm model at the right time, and also to make sure that it can address the question that is being asked of the research. What are you trying to mimic, and is what you're doing appropriate to mimic that situation? Especially when you want to do, say, a compound screen, go for an early in vitro model to compound screen before moving on to further, more advanced models that are also more costly and time consuming as well. And ultimately, what we need to do is to create and agree on standardized models for testing biofilms, for assessing claims of anti-biofilm activity, not just antimicrobial, anti-biofilm activity. And what that is going to require is the creation and maturation of global biofilm networks, and that's going to be key to doing that. And there are a number of centres, such as the National Biofilms Innovation Centre in the UK, Montana State Biofilm Group, Skelsey in Singapore, as well as Kosterton in Denmark, and a few others as well, which are international leaders in that area. And what that ultimately will also have to entail is the collaboration between academia and industry to achieve these goals, to form standards groups to make this a realization. And these will be the goals that need to be done to change the translation of research and making sure that what we're doing successfully translates from the R&D and academia or industry through to an actual efficacious product on the market. So with that, I'd like to thank the members of my lab, Miguel Camara, Manuel Romero, Fadi Sukareth, and my collaborator, Rhiann Griffiths, for the mass spectrometry work, the facilities at the University of Nottingham, and also our funders. Thank you very much for listening.